here today with Dr. Charles Wykoff. We're going to talk about the exciting phase 3 results from the Faricimab study on diabetic macular edema. Charlie, what were the results?
Carmen, great to be here with you. You know, we've been living in the anti-VEGF era for a long time, and I think there's been a lot of pent-up energy in the whole field to try and find additional pathways and additional mechanisms of action that bring value to our patient. And I think the most promising shot on goal there for a number of years has been manipulation of the Tie2 pathway. It's a complicated pathway. There's a lot of different ways you can manipulate that pathway, but now we have strong data showing value with inhibition of angiopoietin-2 having additive benefits beyond VEGF-A blockade. So it's exciting. It's exciting to see this data. As you referred to, we presented top-line data from the Faricimab Phase 3 program, both in DME and neovascular AMD at Angiogenesis this last weekend. And the data is strong. There's good durability signal in both the DME and the neovascular AMD data set. I presented the DME data and Jeff Heyer presented the neovascular AMD data. So happy to talk through sort of the specifics of what we saw in DME. But the short answer is, I think it's exciting. There's still a lot to learn from this data. There's still additional long-term data that will be forthcoming and a bunch of additional analyses of the current data set. But the first look at this data is that it appears strong and meaningful for patients.
What about visual acuity data? Talk to us something about the primary endpoint.
Yeah, absolutely. So first of all, trial design. You know, the phase 2 trials, there were 3 phase 2 trials of Faricimab, 1 in DME and then 2 in neovascular AMD. The DME phase 2 was a very well-designed trial, over 200 patients. The comparator there was ranibizumab, 0.3 milligrams compared to Faricimab. And in that phase 2 program, there was a superiority signal of Faricimab 6 milligrams compared to 0.3 milligram ranibizumab visual acuity wise, with supportive secondary endpoints across the board anatomically. But in the phase 3 program, there was a different comparator that was used. And this, I think, was a really important step to use aflibercept as the comparator in the phase 3 program. So they had the strongest possible comparator arm, aflibercept 2 milligrams given on label Q8 weeks after 5 monthly doses in DME. And then there were 2 Faricimab arms. There was a fixed every 8-week dosing interval, just like aflibercept, except instead of 5 monthly doses, there were 6 monthly doses with the aflibercept arm. And the reason for that 6th dose was simply the phase 2 data. Whereas in phase 2, it appeared that you continued to have visual benefit by continued monthly dosing through 6 months. And so we wanted to try to maximize that opportunity for visual gain. So 6 monthly doses in the Faricimab Q8 arm. And then the 3rd arm of the phase 3 trial was Faricimab PTI, our personalized treatment interval, which was essentially a treat and extend application of Faricimab after 4 monthly loading doses. So we're 3 arms in both Yosemite and Rhine, both independent but identically designed phase 3 trials. And these were structured as non-inferiority trials, where the first sort of statistical look was non-inferiority of each Faricimab arm compared to the aflibercept arms. If that was met, the statistical plan was to then look for superiority among the treatment naive population. Because previously treated patients were allowed into the study, but that was capped at 25%. And non-inferiority was achieved, but superiority, visual acuity wise, was not achieved. So what I mean by that is that through 1 year, the visual acuity gains were robust across all of the arms, with gains of between about 10.5 to about 11.5 letters across all of the arms, with the 95% confidence intervals all overlapping. So no indication of superiority and clearly non-inferior between each of the arms, Faricimab arms compared to aflibercept. Where the value of Faricimab really appeared to shine was both from an anatomic perspective and from a durability perspective. And in particular, we reported 5 anatomic endpoints, right? So if we look from a CST perspective, change from baseline across the studies appear to consistently favor faricimab. The faricimab arms, both of them appear to be consistently drier in the aflibercept population. The second anatomic endpoint that appeared to favor faricimab was looking at the proportion of patients that had absence of DME. And this was defined as a CST below the enrollment threshold of 325 microns using a spectralis. And this proportion was consistently greater with faricimab, reaching over 80% within 1 year. And the third point that favored faricimab was the proportion of patients with absence of intraretinal fluid. And this was consistent across week 16, 48, 52, and 56. So those three anatomic endpoints clearly favored faricimab. The other two were very similar between the comparator aflibercept and faricimab, and that was absence of subretinal fluid. This was achieved in the vast majority of patients, over 95% of patients in each arm. And then DRSS improvements, diabetic retinopathy severity improvements, again, were similar between the arms. And the last point I would make is that anatomic data was very consistent with the strong durability signal that was seen. So 12 weeks or greater dosing was achieved within 1 year in about 72% of patients. And 50% of patients were able to make it 16 weeks between faricimab dosing by the end of 1 year. So clearly a signal for a durability and clearly a consistent anatomic signal with faricimab.
So how do you think this faricimab will be deployed clinically as we see it being used? I presume this is going to receive FDA approval. How will it be used?
Yeah, it's a good question, and you bring up a good point, right? It's not yet FDA approved. It's not yet commercially available. I'm not involved in that process, but the company, the sponsor has said publicly, Roche Genentech, that they are planning to file with regulatory agencies both in the US and ex-US by the end of the second quarter 2021, which seems remarkably fast, very impressive timeline, given that this data has just been publicly released, here in the second week of February. So the other angle to discuss related to commercial utility is of course any side effects, any adverse events from the drugs. And I say that mostly because the environment now is very different, having all of us experience sort of the inflammatory issues with brolucizumab and abicipar in the phase 3 programs, and then once commercial use of brolucizumab began, we saw those numbers validated in the real world. And so the use of brolucizumab has not been nearly what we all anticipated because of the safety signal. So when we look at the safety signals of faricimab, and we've tried to look at those extremely carefully, just given the real sensitivity and the genuine need to understand these safety signals in detail, we noted that the overall rates of intraocular inflammation and endophthalmitis were low across both programs, the neovascular AMD and the DME program. In particular, in DME, if you pool all the faricimab arms and all of the aflibercept arms, the rate of IOI was about 1.3% with faricimab and 0.6% with aflibercept. So numerically, as some have pointed out, that's slightly higher with faricimab. But the interesting observation is that I think all of these rates are low, and below the USPI, the package insert for both aflibercept and ranibizumab in both DME and neovascular AMD. Furthermore, there were no retinal vasculitis cases. There were no occlusive retinitis cases. There was nothing of concern that I saw from a safety signal here. But the good thing related to especially safety and longer term efficacy is that we have second year data from both of these phase 3 programs, DME and neovascular AMD, as well as long-term extension studies. So the sponsor, Roche Genentech, here did a beautiful job of establishing long-term data opportunities early in this clinical trial program. So ultimately, we'll have 4-year data from both DME and neovascular AMD because of the long-term extension studies that are already set up.
Talk a little bit more about the durability data. Could you refresh our viewers about that?
Yeah, this is always an interesting angle, right? Because the comparator arm of aflibercept in both the neovascular AMD and the DME program was limited. It was limited in that it was a Q8 dosing interval after monthly doses at the beginning, 5 monthly doses in DME and 3 monthly doses in neovascular AMD. And the reason for that comparator arm is very clear, right? The reason is that you, from a regulatory perspective, you have to have an on-label dosing arm to be able to get a clear indication for a new product. And that's why you have consistent aflibercept dosing throughout these trial programs. And you don't allow the faricimab arm, or excuse me, you don't allow the aflibercept arms to extend the interval beyond Q8. And so there's always a little bit of a limitation because now you have the comparator faricimab, the new product that has one arm in DME, which allows a personalized treatment interval where you can go longer than every 8-week dosing, and you can actually go shorter than every 8-week dosing. In particular, we allowed patients to get individualized therapy based on their anatomic and visual need. So for example, in the PTI arm, they got 4 monthly doses. And then if their CST was less than 325 microns, they extended the interval over time out to 12 and then every 16 weeks. Whereas if they did not achieve a 325 micron retina or thinner, they maintained monthly dosing actually through the end of 1 year, which was about 7% of patients maintained monthly dosing. But then, right, 93% of patients did not need monthly dosing. And in DME and in neovascular AMD, there was, I would say, a remarkable durability signal. So in DME, over 70% of patients achieved a 12-week or greater interval, and over 50% of patients achieved a 16-week interval by 1 year. And in neovascular AMD, those numbers were remarkably similar. So almost 80% of the neovascular AMD population achieved a 12-week or greater interval, and about 45% of the neovascular AMD patients achieved a 16-week interval. So I think we're seeing a real durability signal here. And how that durability signal will play out in real world utilization will be fascinating, because all of these trials have particular criteria that define these treatment intervals. And in the real world, the criteria that we use might be slightly different. And so that transition from clinical trial data to real world will be fascinating once it's commercially available.
So do you think that the faricimab, the bispecific molecule, will replace the anti-VEGF monotherapy agents?
I don't know. I think it's a little early to conclude that. I personally am a fan of more tools in the toolbox. I only see value when additional agents come to market. It's sort of the analogy I use with my patients is blood pressure medications and cholesterol medications. One of the reasons there are so many products that control blood pressure is different patients respond differently to different medications. And I am excited that we're going to have a new mechanism of action to be able to inhibit angiopoietin 2 and restore Tie2 signaling in these eyes. I think it's going to bring value to patients. Now, will all patients need to have Ang2 blockade to optimize their outcomes? Probably not. But I think we don't know the definitive answer to that. I look forward to additional long-term data, additional secondary and post-hoc analyses of these data to really try to understand who are the patients, what are the particular anatomic characteristics that lend themselves to better outcomes with Ang2 blockade compared to VEGF-A blockade monotherapy.
Will faricimab be studied in non-proliferative diabetic retinopathy without CME?
I certainly hope so. And my prediction is yes. I think that's going to be a very important population to study, right? They think the mechanism of action of Ang2 blockade is multifactorial. And there's some great preclinical data to suggest that inhibition of angiopoietin-2 at earlier stages of disease may change the disease course in a way that VEGF-A blockade monotherapy may not be able to do. So that data is really important ultimately to look at. And as far as I know, those trials are not yet ongoing, but I look forward to being a part of those trials over time.
Well, Charlie, thanks a lot for this exciting data. Another chapter is opened in the therapy of diabetic macular edema. Thank you.
Exciting times for all of us. Thanks, Carmen.
Okay.